water and your bio waste while in space?
Two very good icky questions. It's true. It's very important. So the wastewater, yesterday's coffee is tomorrow's coffee, is kind of where it comes down to. So they're currently able to recycle about 93 to 95% of the humidity condensate and wastewater, the urine that's generated. Solid waste is thrown away. You can recover a little bit of water from it, but it's not worth the expense of having to get rid of the microbes and the other undesirables that live inside of that body. But in order to do the Mars mission, we actually have to increase that recovery efficiency a little bit. So several experiments over the next few years are going to be looking at tweaking that 95% water recovery up to 97, 98% with new technologies. Very good question.
So let's thank Dr. Roberts for sharing a little bit about the National Lab. So our next researchers are part of the Rodent Research 19 experiment. The goal of Rodent Research 19, also known as RR19, is to investigate a proposed method of preventing bone and muscle loss both in space and eventually on Earth, kind of what we talked about. So here to share with us is Dr. Se-Jin Lee, professor at Jackson Laboratory in the University of Connecticut School of Medicine, and Dr. Emily Germain Lee, professor at the University of Connecticut School of Medicine and chief endocrinology and Diabetes Connecticut's Children's.
Great, thanks a lot. I'm Se-Jin. I'm the principal investigator for this project, which we're calling Sending Mighty Mice to Space. This is a collaboration between the Jackson Laboratory, the University of Connecticut School of Medicine, and Connecticut Children's, as well as, of course, the ISS National Lab. You've already seen in the video and also from Dr. Roberts that the focus of this project is a protein that we call myostatin, which we discovered many years ago. And we showed that myostatin normally acts to limit muscle growth. And the dramatic illustration of this was that when we genetically engineered mice to lack myostatin entirely, the muscles in those animals grew to be about twice the normal size, hence the name mighty mice. We went on to show that myostatin plays a similar role in other species as well. And so in the first slide here, this shows one example. This is a bull named Hotline, in which we first identified a naturally occurring mutation in the myostatin gene, and you can see how heavily muscled this animal is. The same is also true in humans. In work that we did with Marcus Schulke in Berlin, we identified a child who was very muscular and who turned out to make little or no myostatin as well. So all of this immediately suggested to us the possibility that drugs targeting myostatin, that is blocking myostatin activity, could be useful in a wide range of conditions in which muscle loss or muscle wasting is a problem. So these would include muscle degenerative diseases like muscular dystrophy, but also muscle wasting that occurs in people with cancer, AIDS, heart disease, sepsis, and so forth. Muscle loss, of course, also occurs in people who are bedridden and in the elderly who are oftentimes physically inactive. And one way to mimic that type of muscle loss is in microgravity, as you've heard, where you get the systemic loss of muscle because of the inability to bear weight. So the focus of this project is going to be to determine whether getting rid of myostatin in mice that we send to the International Space Station can prevent or at least mitigate the loss of muscle due to microgravity. And we're going to be taking two different kinds of approaches. One is a genetic approach, so we'll be sending the mighty mice themselves to the ISS. These, as I said, are mice that make no myostatin at all because of the genetic change. But in addition, we're going to be taking a pharmacological approach where we're going to be giving normal mice a biologic, an experimental drug that's capable of blocking myostatin activity and causing the muscle to grow. Now, this particular experimental drug also has another potential beneficial effect. And at that, I'm going to turn this over to my co-investigator, Emily Germain Lee, who will tell you about that.
Yes, so, sorry. This drug doesn't only block myostatin, it also blocks another molecule that's called activin.